Welcome to A-Level Biology Paper 3 revision on what is probably going to be one of the last streams I ever do on this channel, which is kind of mad to say, but I started doing these revision streams all the way back during GCSE season, and now it's been 2 more years. We've worked through AS levels, we're working through A levels. I really can't wait to just be done with it all, to be honest, but we still have a little bit more to push towards. And it's paper 3, which I would argue is probably one of the nicest papers. I think... Hello. How you doing, man? I personally think paper 1 is lovely, with all the content being really nice. It's mostly knowledge. Paper 2 is a horrid paper. And then paper 3 is just kind of like the slack between both papers. I can make some reasoned assumptions here in saying that we're likely going to be getting a lot of practical questions based on the fact that we only got one practical throughout the entire of paper 1 and 2. I can assume that we are going to get a question on immunology. Probably because, we're no immunology questions in paper 1. I can assume we're probably going to get 1 or 2 things on the organism's response topic. So something like osmoregulation, possibly something to do with synapses or receptors. What's this for? This is AQA A-Level Biology. We're going over paper 3, which is happening in 2 days' time. So yeah, I really do hope we get some organisms response questions. Those would be lovely. Yeah, honestly, the kidneys isn't the worst topic, but this is just me yapping, predictions. I'm gonna do some actual revision in just a sec. But yeah, I do hope that osmoregulation to some extent comes up. I hope you guys are all doing well, by the way. I know these streams usually get a lot of people watching and things like that. I hope you guys are doing well. I hope paper 2 went well. I can't lie, I thought it was horrible, but apparently everyone else loved it, which is upsetting. Yes, ADH would be lovely. ADH would be lovely. Oh, really? Wait, it was a chemistry paper today? Or just revision? Man. Oh, I'm sorry to hear. Honestly, massive respect to those of you guys that do chemistry, because it looks like a nightmare. I can't lie. I can't lie in the slightest. Really? Okay. I'll have to ask the people I know that do it. Man. Okay, I'm just gonna pull up the stream so I can actually see what people are saying without glaring into the... Chemistry is evil. It actually is. It is. So in terms of what I think is gonna come up, again, I'm literally just... I'm not a teacher. I can't obviously foresee. I would love osmoregulation to some extent. Wonder if muscle contraction could come up. Earlier I looked at a past paper and it was interesting how it was only 7 marks, but they did a muscle contraction question with some really weird application. So I feel like they'll probably shoehorn in muscle contraction. It's such a big topic, with a lot you can kind of yap about. I realise we got questions on respiration a little bit. We didn't get any questions on photosynthesis as far as I remember. So realistically, they could easily do a massive... 15 marks at the end of the paper. They could easily do that on something related to plants. Thank you, man. Thank you. How to do the essay. Oh, okay. Well, I want to get into that as well, actually, in a tad. I want to get into the essay. I don't know what you guys are predicting or hoping for the essay. I think there are. I think there are. The way we've been told to do it, how long might this be? I'll probably be live for like an hour or two. Basically just till my parents get home. The way we are, I mean, I think the way you have to do it is a lot of people do an intro, and a conclusion. You're not meant to do that. There's no marks to be gained through that. It's literally just, 4 topic areas. And the big thing that I always mess up is not including enough like width with them. So if you got a question on the importance of water, you're not allowed to mention translocation and transpiration because it's the same marking point. So make sure to pick things from, paper 1 and paper 2 content. Make sure that the topics are like pretty far apart from each other. That's my advice, because I've lost a lot of marks in the essay questions by talking about really similar things. The easiest ones, I would say, is try and shoehorn in respiration or photosynthesis. Those are usually like really lovely ones to talk about. Things like co-transport is a lovely one that you can talk about. You can normally apply co-transport in a lot of different ways. Talking about movement of ions, the importance of diffusion, importance of ATP. And please help. In fact, I might just have to make a pin comment. Just one sec. I'm just gonna pin this so that hopefully people realize I'm not qualified at all and I don't know what I'm doing. So what I might do actually, before I look over some past paper stuff, I might just do a handful of essay plans for some things that people think are going to come up. I don't know what could come up. I could look online for predictions, but if you guys have any that you think will come up, feel free to share. I mean, I'll go look at the paper 3 for 2017 and 2018. We'll go see what they had. AQA paper 3. Yeah, we'll go look at the 2017 paper, see what that says. So the 2017 paper had the importance of nitrogen-containing substances or the importance of diffusion. I think that's a lovely choice. That is a lovely choice. I guess we can just kind of like, I don't really need to do it like this. I can just kind of do like a spider diagram. So the importance of nitrogen-containing substances. This would be a really lovely one, I think. You could mention like literally anything. DNA, ATP, mRNA, and then obviously you're able to waffle about those topics a lot. Also, nitrogen-containing substances, you could talk about enzymes in terms of, they're comprised of amino acids. So I feel like you could easily shoehorn in a lot of really easy topics here that could get you up to like 4. I would say you could talk about DNA replication, because it says the importance of it in biological systems. So obviously the importance of DNA replication in the process of mitosis. I wouldn't really know what else to mention in terms of importance, but obviously that would be your big over one, is cell replication, linked to what cell replication does. Protein synthesis. That would be a lovely one to write about for nitrogen-containing substances. You could yap about its role in leading to, the different polypeptides. You could give it like a handful of examples. That would be a nice one. I don't know what you could talk about for ATP. I guess any of the processes, and just make sure to refer to the role of it. Because ATP is a nitrogen-containing substance, so I guess the role of ATP in co-transport, but then obviously heavily discuss the role of ATP though. And then what else? I guess literally just enzyme control reactions. You could do a whole thing on literally just the role of enzymes. And I think that would be enough to get you to 4 different talking points. I think. Yeah, I think that would be decent. And then in terms of the importance of diffusion, this would be, I mean, you could talk a lot more broadly here about a lot of stuff. You could talk about synaptic transmission. That would be a lovely one. Really easy. You can talk about the diffusion of sodium ions, calcium ions, and obviously what that does. Again, it's always talking about the importance, so talk about why that's important. Co-transport, again, would be a lovely one to talk about. One of the easiest processes by far. Probably, I'll try and, like, do a... it just shows how you structure. Okay. I'll do that in just a sec. I can't lie, like, my structure is basically just describing the process word for word, and then at the end and the beginning of each paragraph, I'll briefly... That's where I lose marks, because I only briefly talk about its importance, but I will. So synaptic transmission, co-transport. What else could you do for diffusion? My head has gone blank now. What else could you do for diffusion? I guess you could do the light-dependent reaction and the role of the hydrogen ions, because they obviously diffuse down the gradient. And I guess if you really wanted to be like, you really had nothing else to talk about, maybe you could talk about oxidative phosphorylation. Because they're both really similar, so you could probably, like, squeeze marks out. Obviously, the importances is the bit that everyone kind of fumbles on. Talk about the importances, and you obviously have to link it to the topic. So when talking about the importance of diffusion, in synaptic transmission, that's going to be the importance of diffusion of the calcium ions and the sodium ions. Then obviously you can refer to the fact that the importance of the calcium ions is it allows the vesicles to fuse with the membrane. The importance of the sodium ions is obviously going to be that it allows the continuation of the depolarization. And obviously you have to refer to it as diffusion. Because that's the thing that I think a lot of people kind of, like, overthink, possibly, is when it's talking about the importance of, they'll talk about the importance of co-transport. They won't talk about the importance of diffusion in co-transport. I think that's where everyone goes wrong. Because, I mean, look, like, most of us can, like, pretty clearly write about the process. But it's kind of a extra bit that they want. I have no idea. So that would be, like, again, I'm obviously not the best at these essays. In fact, I might have, like, a copy of one lying around. Hold up. Got like a Polly Pocket here. I don't know how many marks I got off of this. Okay, I only got, well, I say only, I got 19, which was fine. The only feedback I got for this... Wait, what question even was it? Before I start yapping. The importance of responses to changes in the internal and external environment. So I'll quickly kind of bullet point what I did. I wrote about... blood glucose regulation. I wrote about hemoglobin. These are horrible topics to write about. I don't know what I was thinking. Reflex arc. What are these topics, bro? I picked the most, like, horrible topics. And speciation. Yeah, no wonder I thought I fumbled this. Okay, so those are the topics I wrote about. Now, obviously, you can, you can easily write about a lot of these. You can easily write about the role of blood glucose regulation. The bit that I kind of had to work on, my teacher said, is literally the idea of, like, I mean, it's kind of, it sounds a bit stupid me saying it out loud, but literally reinforcing the importance. So what my teacher said is, for blood glucose regulation, I talked about how insulin works, beta cells, things like that. What I didn't go on to say is the effect of not having blood glucose regulation would have. So it's this idea of, if there's too much glucose, water will leave the cells, via osmosis, leads to cell pronation. So it's literally the idea of, you talk about the process itself, that's like your AR1, but then you have to go in and basically describe the significance of that process. And then what my teacher makes us do, at least, is like allude to what would happen if that process wasn't a thing. So for blood glucose regulation, if you didn't do that, what would happen? You could easily link, like, when talking about the reflex arc, that would be a lovely one to talk about the importance of, because then you could say it helps organisms avoid predation, avoid harm, and kind of elaborate on that. Like, what did she say for mine? Reflex arc. Yeah, she literally just said avoiding predation. So, in terms of, like, AO1, it really isn't that bad. It literally is just adding those extra little bits at the end of each paragraph where you're talking about the importance of. Like, the way I will literally do mine, the way I'll literally format mine, and it's what a lot of people in my class will kind of do as well, is you'll do a whole section on AR1, and then a tiny little, like, ending bit talking about its importance. And I don't know whether this is necessarily a bad thing. That's the way I do it, and I get decent marks. But, yeah, it is definitely something worth noting, is you don't have to embed AO2 within the paragraph. You can just add a little bit at the end, like, talking about the importance of. And if you can't figure out the importance of something, just really make it like a bare bones answer. Like, without synaptic transmission, what would that mean? Like, things like that, really. I'll do like a handful more essay plans, and I'll try and emphasize more of the AOT stuff if I can. What was the 2018 paper? What was it? The importance... Oh, what the hell? The importance of the control of movement in cells and organisms, or the importance of interactions between cells and between organisms. Huh. Those are really odd ones. The control of movement in cells and organisms. Am I overthinking that, or is that... Huh. Control of movement. Let's see. I always... A good thing that a lot of people do is they'll flick to the essay question first and just see what it says so that they can think of stuff during the exam. I'll definitely have to do that. Because I'm struggling here. The control of movement in cells and organisms. So is this going to be talking about diffusion, osmosis? That's a really odd one. Huh. Am I actually being dense here, or like... Maybe I am. Maybe I am being dense. Huh. I might need to check the mark scheme here to see what the topic points are. I mean, the first one that came to my mind was muscle contraction. Because then, for the A or 2 of that, you could talk about muscle contraction being used to, evade predation, things like that. But, oh man, that's a really ambiguous title. And I don't like that, actually, because they've given me way too many options. Let's see what the mark scheme says. Okay, the control of enzyme action. That's an odd one. Oh, mitosis? Okay. Transport across membranes, that would definitely be one. Cell recognition and the immune system. Receptors, that would be a nice one. Muscle contraction, control of water potential. Okay. Okay then, that's not a particularly bad essay. Just really ambiguous. And as for the other one, the interaction between cells and organisms, the first one that I thought of was, the immune system. Like cell recognition, things like that. What does the question say? The importance of interactions between cells and between organisms. That is a really odd title. Because I'm struggling to infer what that implies. Interactions between... Oh, okay. So things like, for active loading of translocation, things like the companion cells. Okay, so then you could refer to ADH, possibly, for that one. Translocation. Okay, that's not a particularly bad title. Yeah, control of water potential. Okay, cool. That's not too bad then. Okay, we'll go look through some more. 2019. I have a feeling this one's going to be even worse. Let's have a look. Oh, what the hell? The importance of DNA as an information-carrying molecule and its use in gene technologies. What kind of question is that? That's horrid. I mean, obviously the most obvious one's there. is going to be for the bit about information carrying, transcription and translation. Then the importance of that is going to be talking about the idea of the bases, the base sequences code for different amino acids, leading to the different expression of proteins. That would be a lovely thing to talk about. Then possibly you could refer to DNA replication and the idea that DNA needs to like remain perfectly copied. Maybe refer to the fact that if it doesn't, it leads to uncontrolled cell division. Then, in terms of gene technologies, the use of DNA in gene technologies, is this going to be the role of transcription factors, possibly? That's the only real one I can think of. Or is this going to be like cloning of DNA? That's such a horrible question. I would never think of doing that, to be honest. Then the question next to it is the importance of bonds and bonding in organisms. I feel like that's a way nicer one. Because I guess a whole paragraph could be talking about condensation and hydrolysis and the importance of that. One of them could be translation. The idea that there's the condensation reaction between the amino acids forming the peptide bonds, and you could talk about the role of ATP in that. The role of bonds, possibly hydrogen bonding, when talking about transpiration in the xylem. That would be a lovely one. And then, what else? Oh, maybe just talking about ATP. And when ATP bonds are broken, the energy release, talk about the role of ATP. That would be a lovely essay. Importance of bonds and bonding in organisms. Yeah, that would be a lovely essay. I can't see anything wrong with that title. Way nicer than the latter. Okay, we'll continue. The 2020 paper. The importance of enzymes and their importance in... That is crazy. That is a crazy title. Okay. I would guess you could probably just write a whole paragraph about enzymes themselves. The idea that the temperature needs to remain at an optimum. Talk about the induced fit model. And then obviously the whole gimmick with enzymes. Talk about reducing activation energy, leading to increased metabolic reactions. Things like that. That could be a lovely paragraph. Enzymes are very important. Possibly the role of coenzymes for things like oxidative phosphorylation. That's where NAD and FAD donate their hydrogen ions to the transport chain, right? That is what happens. I'm not misremembering that. Yeah, they donate. Obviously they donate electrons to the electron transport chain. Yeah, that would be a lovely one. And then finally, what else can we talk about for enzymes? Possibly the role of Ribisco in the light-independent reaction. I think. Because, yeah, that's the importance of enzymes. Yeah. Talk about its role in combining IUBP with CO2. Talk about what that leads to, and obviously that's your importance. Oh, possibly, yeah. I don't think that would be vague. I think you would probably just have to like hammer down a specific type. Like the nicest one to talk about would probably be like lipid digestion and the role of lipase. Um, and then obviously link that to what happens afterwards with like micelles, possibly an absorption. And then I guess you could then link that to like lipids itself. I don't know. I don't know. I don't think that would be too vague. That would be a nice one. I'm trying to think what else you could talk about. Um. Oh, things like the second messenger model. I think where you have like adenal cyclase. Oh, active phosphorylase as well for the effect of ADH, where that causes the aquaporins to. And yes, true. To be fair, I can't stand that topic. It would be a nice one to talk about, but honestly, I hate that topic so much. I would probably avoid it. Yeah, I think that second messenger model would be nice. And then the next essay title is a horrid one, I think. The importance of variation and diversity in organisms. I wouldn't even know where to start with that one. The importance of, what was it, variation and diversity? I would have no idea how to even link those to like an AO2. I mean, the only one I could literally talk about would be speciation. But I wouldn't know how to talk about the importance of that. The only thing I would be able to say is it increases. How would you even word it? Yeah, talking about like advantageous alleles being passed on, um, leading to population growth. Yes, actually, that would be a. But honestly, that would be the only one I could think of. Could you even talk about natural selection? I mean, possibly. Thing is, like, I wouldn't know what else to talk about. Like, if I got this question, I would sob. Especially if a choice was between something equally worse. I want to see what the mark scheme says. Oh, that is true actually. Yes. For natural selection, most likely, because then you could say, you could give an example of like, I don't know, like antibacterial resistance and like the idea that that's because of mutations and natural selection. I want to see what the mark scheme says, because I. Oh, okay. Huh. One of the mark points is slow and fast twitch muscles. What? Oh. Ah. Wait, yeah, that does kind of make sense, because it's variation in organisms, like two varied types of. That is weird. Obviously, genetic diversity in meiosis, genetic diversity in mutation, genetic diversity in adaptation, speciation, yes. And another one, just for talking about proteins having a variety of functions. I guess, yeah. Oh, yeah, it makes sense looking, but no, like, that seems like such a risky one to put in. I can't see anyone putting that. The one about proteins does make sense though. Like, obviously, different proteins for different functions. But, again, would be an odd one to write about. Definitely would not be my ideal paper. I'll move on to the 2021. Again, basically all I'm doing is looking at the essay titles. Oh, take care, man. Thank you for watching. I appreciate it. And good luck, good luck. The importance of okay, this is a lovely one. I think. The importance of complementary shapes. Obviously, the first one that comes to mind would be, literally just talking about enzymes. That's definitely the easiest one to get an AO2 out of as well. Because when talking about enzymes, you can link it easily to the function. The idea that it reduces activation energy, it catalyzes the reaction, increasing the rate of reaction overall, and talk about the importance of what that means. What else could we do for complementary shapes? I would probably say something like synaptic transmission. The idea that the neurotransmitter binds to the receptor specifically, what that binding means, and then maybe even you could talk about drugs. The idea that some drugs will mimic the shape of neurotransmitter, leading to increased depolarization, or you could talk about the role of drugs in blocking the receptors. That would be a nice one, I think. That would be a lovely one to get an ALT out of. What else could you do for complementary shapes? Complementary shapes. Would you be able to talk about things like DNA replication here? Because it's complementary bonding, or is that something different? Let's have a look. Let's have a look at what the mark scheme says. Yes, DNA replication. The importance of complementary shapes. And obviously with DNA replication, that would be, again, a really nice one to talk about the importance of. You could easily talk about the importance of DNA replication and the idea of complementary shapes. Again, the big mistake that I often make with the essay questions is I'll talk about the importance of a process, not the importance of what the title is. Oh, thank you, man. So, instead of talking about the importance of DNA replication, it's the importance of complementary shapes in DNA replication. That's the idea, at least. So obviously the idea that the bonds are complementary, the enzymes are complementary, the base pairing, that would be a nice one. What else could we talk about here? I don't know. Yes, I am a student. I'm not a teacher. I'm trying to think what else you could talk about here for complementary shapes. There'll be some obvious ones, but... maybe the immune system? That feels like quite a broad thing to talk about, but the idea of the, like, possibly just talking about antibodies, that would be a great one. Talking about the antibody-antigen complex, that would be a nice one. So that's a pretty decent essay. Like I would be happy if I got a title on that. I'll go look at the 2022 paper. Which is... oh, I've seen this paper before. I've done this one. The uses and importance of ATP. Yes, that would be a nice one. Because that's basically just saying the uses of active transport. Obviously, for one mark point, you could just talk about ATP itself, the properties of ATP, the structure, and the importance of its structure. That would be a really nice one. Then you could talk about co-transport. The idea that ATP is used during the sodium-potassium pump. Talk about the idea that it's ATP which causes the pump to have an affinity. That would be a nice one. What else could we talk about for ATP? The role in active loading. For translocation, that would be a lovely one. Talking about how the energy is used in order to pump the sucrose across. Finally, what else can we talk about for ATP? If I was really ballsy, I would talk about the sliding filament hypothesis. I personally won't because I suck at that topic and I don't want to write about that. But that would be a really nice one to talk about. What else could we talk about for ATP? Oh, maybe. Oh, okay. This would be quite a ballsy one. Maybe the role of ATP in glycolysis? Because obviously ATP is used to phosphorylize the glucose. That would be an odd one to write about, but I feel like they'd allow that. Yeah, I feel like they definitely allow that. Yeah, okay. That would be a decent one, definitely. The next one is the importance of cycles in biology. That is an interesting one. I've already thought long and hard about this title. I know which ones I would definitely talk about. Obviously, meiosis would be a lovely one to talk about in terms of AO2. You could talk about the increase of genetic variation and what that means for survival, everything like that. That would be a lovely one to talk about. Importance of cycle, definitely the Krebs cycle I would talk about. The importance of that is obviously in providing, well, not only providing some ATP, but its overall role of providing NADH and FADH. Obviously, the importance of that being that it leads to ATP synthesis on the electron transport chain. The Calvin cycle, its role in leading to the creation of pentose sugars. Lovely one. And I don't know what else you could talk about in terms of cycles. There's definitely some nice ones. Mitosis, possibly? I think mitosis would be a bit of a last straw. Like, if I really couldn't think of anything else original, definitely mitosis. But there's definitely some others you could talk about, I'm sure. Trying to think now. Maybe co-transport, because co-transport is a cycle where concentration gradients change. That would be a nice one. Definitely a nice essay title. Let's look at what the specimen paper says. The importance of movement in living organisms, or the importance of receptors. Okay, receptors. This would be a very easy choice for me. The importance of receptors in organisms. You could easily talk about, I guess an obvious one would be immune response. the idea of the formation of the antibody-antigen complex. Obviously, another would be synaptic transmission. Then, honestly, actually, I wouldn't know what else to talk about here for receptors. Possibly the mechanoreceptor, the idea that increased pressure leads to increase of depolarization. Maybe photoreceptors, the difference between the rods and cones. Okay, that wouldn't be a bad one. That wouldn't be a bad one. I quickly want to go over two that I've heard people talking about. The first is the importance of water in organisms. And honestly, I do hope that this one comes up. It would be a nice one to talk about. Obviously, a really nice one to just discuss would be the properties of water. Because you could talk about, I mean, the most obvious one I would go with is water's high heat capacity. Oh, thank you, dude. The high specific heat capacity of water, you can talk about that in terms of its role in enzymes, maintaining an optimum temperature, what would happen if that didn't occur. That would be a nice one. Then I would probably go for osmoregulation. The idea that if this doesn't happen, you know, what the consequences of that, water loss, things like that. Then I would go with probably transpiration. You could easily link that to the cohesion tension theory, talking about water being adhesive to the walls of the xylem, the idea that water remains in an unbroken column because of the hydrogen bonds. Then what else could we talk about for water? Possibly the idea of condensation and hydrolysis. The idea that water molecules join together the bonds. Maybe tissue fluids. Oh, and obviously for osmoregulation, you can talk about the role of ADH. That would be a really nice marking point. So yeah. Okay. So that's basically everything for like the essay questions. What I want to do now is I want to go over a handful of like the practical stuff that could come up. Because obviously, paper 1 and 2 did not have any practical stuff really. The only required practical we got was the choice chamber, which I thought was a really nice question. I think, I don't remember. So let's have a very brief look at a handful of the non-required practicals, because these could come up too. The most obvious ones are things like the Benedict's test, the emulsion test, the starch test, the proteins test. Those are all really nice ones that you could definitely get asked about. Cell fractionation. This is a really lovely one. You could literally, you get so many marks for talking about homogenization when it's really like one of the easiest practicals to yap about. It's this idea that first of all, you homogenize. Then you usually get 1 or 2 mark points for talking about the solution, where it's cold, isotonic, and buffer. Then it's this idea that you centrifuge it. Oh, and obviously additional mark point for talking about filtering debris. Then you centrifuge it at a low speed to separate the nuclei. And then finally, you respin at a faster speed to get your pellet of the desired organelle, i.e. chloroplast. You could easily get asked a 4 or 5 mark question on the role of homogenization and centrifugation. It's a lovely question. I hope it comes up. Then in terms of other ones, aseptic techniques. This is such an obvious one, but you could get asked 4 marks for this. I've seen plenty of questions on aseptic technique. The most obvious ones is going to be things like wash hands, flame the neck of a bottle, work near a convection current to kill any airborne pathogens, use a sterile pipette. Literally, you can get up to 4 marks just by yapping about aseptic techniques. It's really not that bad. Then there is chromatography. This is such a lovely one. I will draw a big ass thing here. Chromatography of photosynthetic pigments. This is lovely. First thing is draw the line in pencil. You always get a mark point for saying that. Because remember, pencil is insoluble, pen, ink would be soluble. You would obviously add your chlorophyll pigment. If you get asked about a pipette, mention a pipette. And you make sure to add the solvent below the origin line. Obviously, so that the pigment doesn't just immediately dissolve. And then the way you confirm that this practical has kind of needed to be stopped is once the line itself reaches close to the top. Then obviously you mark your solvent front, you calculate the RF value. It's this idea that, let's say we have, oh, what the fuck? It's this idea of if we have 2 chromatography paper in 2 different solvents, A and B. If this one has if this one has 4, this one has 6, why would that be? It's this idea that the chlorophyll is more soluble in this liquid, or a certain pigment is more soluble in that liquid. Then everyone's favourite practical is talking about quadrats and using those to estimate the number of something. You can also use quadrats to estimate the species richness, to estimate a percentage cover, anything like that. Like literally, you can apply quadrats in so many different contexts. If we have a 200 by 200 meter field, you are going to get 3 mark points for saying 3 easy things. The first is saying that you need to use random sampling to generate a grid of coordinates. It's this idea that it removes bias if you use random sampling. Then it's this idea that you use a large sample size using quadrats. The larger the sample size, the more representative of the whole area it is. Then finally, it's this idea that you do whatever math needs to be done. So if you're calculating percentage cover, if 20 out of 50 quadrats had a plant in, your percentage cover is 40%. Okay, cool. Then there are the actual required practicals themselves. Things like the respiration practical. I'm trying to even think of which ones there are. The respiration practical likely isn't coming up. One which could come up, and I know that everyone hates, is the DCPIP practical. I don't even remember how you draw this one. This is the one where DCPIP acts as an electron acceptor. So you get your photosynthetic pigments or whatever, your chloroplast, your plant homogenate, you get whatever it is, you pop it in the tube, you put DCPIP in. Then you can tell when photosynthesis is occurring because obviously you'll get a visible colour change. You could get asked about that. The idea that the colour change is subjective, it's it's qualitative, non-quantitative. This idea that using colorimetry would standardise the test. So the DCPIP practical isn't that bad, I don't think. You could get asked it in a variety of contexts. We haven't been asked once a question on photosynthesis in paper 1 or 2. That leads me to believe this is going to come up. I feel like this is definitely going to come up in some capacity. Maybe not this practical entirely, but definitely could come up. I mean, in the past, there's been a question on the pitometer, which is how you check for rate of transpiration. It's this idea that, like, you attach the plants, and then it measures the rate of water uptake by the plants. So we could definitely get asked either of these. The rate of water uptake would be an interesting one. DCPIP would definitely be a nicer one though. Okay. Well, I have been live for basically an hour. I don't know whether I'm going to head off or just kind of keep yapping. But I mean, like, yeah, I feel like we should be fine, you know? Wow, 124 viewers is crazy, by the way. Thank you, guys. That's cool. Okay, what else do I want to do? Because honestly, I've revised for like 2 hours today, my head's gonna explode. So let's very quickly. There are 3 things I want to quickly go over before we wrap up. One is, in fact, no, we won't even go over them properly, I'll just talk. First one I want to go over is the idea of just the pathogens topic as a whole. The ELISA test. I love this practical. Well, it's not a practical, it's just a test. I love yapping about this because you could even refer to it in context of receptors, anything like that. This idea that at the bottom of a well, you have something like HIV antigen. Hello. You have the antigen, you then add the sample, and if they are positive, it's going to contain an antibody. Then this is where it kind of breaks up depending on what context you're being asked about. What do you mean? What do you mean? TG Wards? What do you mean by that? This is where the test varies depending on what is happening. In some contexts, you will then add another antibody, which will bind to the current one. You will add a substrate to that. You'll add an enzyme. It really. Oh, okay. It honestly, it just depends. It depends on what context. Man, I have no idea. I'll be completely honest with you. No idea. So yeah, that's where it could very much vary when talking about testing for some things. It could definitely come up. Like it's come up a lot in the past. It could definitely come up again. I hope it does, because that would be a really nice one to talk about. The idea of the antigens binding, the importance of washing out a well in order to make sure that the unbound antibodies are washed off. Then, of course, everyone's favourite is phagocytosis. Yes, we're doing biology. Everyone's favourite is phagocytosis. This is a lovely one. Thing is, I always end up getting this one slightly incorrect. No, don't worry about it, man. Don't worry. I was like, oh god, what have I missed? It's this idea that the pathogen, again, incredibly crude drawing here. The idea that the pathogen is engulfed by the phagocyte. You usually get a mark just for talking about that. The idea that it is placed into a vesicle. Again, that could be a mark. The idea that it then fuses with the lysosome, obviously leading to a phagosome. Then the idea that the hydrolytic enzymes are released. Always mention endocytosis. It's a lovely mark point. They destroy the pathogen, digest it, and then the extra mark point is that they become antigen presenting. They present the antigen on the cell surface membrane. And then that is where you can then link this into a lot of other contexts. You have the antigen presenting phagocytes. Then, obviously, you have the helper T cells. When they bind to the antigen presenting cell, they're going to undergo... Yeah, we'll do computer science. Have you finished computer science now, by the way? I realise I forgot. The helper T cell undergoes cell division after binding. And then this is where you can waffle about a load of different processes. Oh, okay. Oh, it is. Okay, cool. That's cool. That's where you can then link it to the cell-mediated response. This is the easiest one to talk about. It's this idea of the cytotoxic T cells. Helper T cells will bind, or will activate. They undergo mitosis. They release perforin, which is an enzyme that literally perforates the cell membrane, causing it to become freely permeable. So if we draw a very crude bacteria, it will literally perforate the membrane. Yeah. Meanwhile, with the humoral response, this one is a tad more fiddly to remember. It's this idea that the helper T cells will activate plasma B cells. Then these plasma B cells will undergo mitosis. Some of them will remain as plasma B cells. Some will go on to be memory B cells. Then it's this idea that the plasma B cells will release a specific antibody which has the same shape. It's complementary to the pathogen's antigens. The antibodies will bind and they will either neutralize or agglutinate to the pathogen itself. And then memory B cells remember the specific antibody, the specific antigen, and that's useful for reinfection because it triggers a more rapid response. Realise Declan's not a mod in here. There. So yeah, that is immunology. Obviously, then you can talk about vaccines, the role that they have. Possibly a 5 or 6 marker on vaccines would be lovely. The role of the ELISA testing, that would be lovely. Like, really, you could get asked a lot. There's only 2 more things I want to go over, then I'm going to leave it for today. First one is photosynthesis. It's easy enough to forget different processes in this. It's the idea of a light-dependent reaction. You have your chlorophyll. You have the thylakoid membrane. It's the idea that photons will excite electrons in the chlorophyll, causing electron to be excited, and it travels down the electron transport chain. While it's traveling down here, it's going to release energy because it's undergoing a series of redox reactions. Then we have photolysis. This is where the light energy is used to split a water molecule. The water molecule becomes split into, I don't remember the way this goes. Oh, hydrogen and oxygen. And then the hydrogen becomes a proton and an electron. The electron obviously goes to replace the one lost in the chlorophyll. The proton, or the H plus ion, remains here for now. Country, England, the UK. So the energy given off by the redox reactions establishes a proton gradient, the electrochemical gradient. The hydrogen ions are going to be moved up here. Obviously, there's a greater concentration of them up here, so they are going to move back down using a channel protein. This channel protein is ATP synthase, which converts ADP and PI to ATP. So what we get from this is ATP, and the hydrogen is collected by NADP. So the H plus is collected by NADP, becoming reduced. Then this links into the Calvin cycle. This is where your 2 products from the light-dependent reaction combine. They're used. So what you have, and this occurs just in the stroma. Can you repeat? Yeah, okay. What happens is your 2 products from the light-dependent reaction, ATP and NADPH, they are used in this cycle. What it is you have, I think it's ribulose biphosphate, something like that. I'm not a teacher. RUBP combines with CO2, and that's catalyzed by the enzyme Ribisco. That leads to the formation of an unstable 6-carbon compound, which quickly breaks down into, I think, GP? It's either GP or TP. TP. Yeah, that's crazy. I'm not that old. Where was I? Oh, it was GP first. Sorry, I read too far along. I do not teach chemistry. I'm not a teacher, and which grade? I'm not American, so I wouldn't know which grade it correlates to. I'm 18, so whatever that is in terms of grades. No, I'm not. Stop saying that. That's misinformation. You prick. Sorry, it's GP first. Then, because of ATP and the oxidation of NADPH, it becomes TP, which then leads in to the rest of the cycle, where 5 carbon are reused, 1 carbon goes off to be a... Is it a hexose or a pentose sugar? Pentose. So yeah, that's the Calvin cycle. I hope it comes up in some capacity, because it's lovely. It's literally just the role of Rubisco in catalyzing the reaction, 2GP, 2TP, the role of NADPH, the role of ATP, boom. Lovely cycle to talk about. And you could apply it in a lot of different contexts. When talking about the role of ATP, when talking about the role of cycles, when talking about the role of enzymes. Like, literally, so much you could talk about. And I think that's probably where I'm going to wrap things up here. I mean, I will definitely take a further look at some more stuff tomorrow, the day before the exam. I don't recall anything else that I really want to revise. Probably worth giving a look over speciation. I think that would be a good... In fact, I'll write this down quickly. Giving a look over speciation would be an easy one. Right, that is crazy. Osmoregulation. I want to go over that tomorrow. I'm okay with ADH. Right, that is a crazy thing to ask. What else? Oh, obviously... Right, that is... Stop spreading misinformation. Fuck off. You're actually insane, you know that. Myogenic control, that did come up in paper 2. So I'm going to go out on a limb and say we don't need to revise that. Muscle contraction, I'll go over that. Then, in terms of anything else to go over, it might be worth going over translocation. Might be worth going over transpiration. Right, that is insane. That is actually wild. I'm gonna go kill myself now. Anyways, final thing is most likely these going over, and then I'll probably also, just as a tiny thing tomorrow, go over the mass flow hypothesis, transpiration stream, possibly hemoglobin, and then obviously we're going to have to take a very quick delve into things like transcription factors, methylation, and gene technologies, because they could definitely come up in some capacity tomorrow. Wednesday. So yeah, that's all that I really have time for. I hope that you guys are doing good with revision. I'm attempting to, definitely. Take absolute care, guys. Hopefully, I'll catch some of you tomorrow. And yeah, that's basically about it.